Responses to the extracellular environment involve receptors that transmit signals to second messengers. Transmembrane signaling is accomplished by only a few mechanisms. First are ligand-gated ion channels. They are transmembrane proteins that act as ion channels. They open or close upon binding of a ligand. A well-known example is nicotinic acetylcholine receptor. Second are G-protein couple receptors. They are transmembrane proteins. Binding of a ligand stimulates a GTP binding protein called G-protein, which in turn generates an intracellular second messenger. Well-known examples include glucagon receptor, adrenergic receptors. Third are enzyme-linked receptors. These receptors are transmembrane proteins with intrinsic or associated enzyme activities. Binding of a ligand allosterically activates the enzyme activity, which typically affects phosphorylation of a target protein. Examples include insulin receptor, EGF receptor, and growth hormone receptor. Fourth are nuclear receptors. They are intracellular receptors. A lipid-soluble ligand crosses the cell membrane and binds the receptor, which typically control gene transcription. Example includes glucocorticoid receptor, retinoid receptor. These four classes of receptors are summarized in this slide. Today, we will focus These four classes of receptors are summarized in this slide. Today, we will cover 6 topics. We will start with ligand-gated ion channel. Ligand-gated ion channels are a group of transmembrane ion channel proteins which open to allow ions such as sodium, potassium, calcium, or chloride to pass through the membrane in response to binding of a ligand, such as a neurotransmitter. Ligand-gated ion channels are classified into 3 superfamilies. One is cyclic loop receptors. Second is ionotropic glutamate receptors. And third, ATP-gated channels. Cysteine loop receptors have a characteristic loop formed by a disulfide bond between 2 cysteine residues in the N-terminal extracellular domain. A well-known example is nicotinic acetylcholine receptor, as shown here. It is a pentamer comprised of 2 alpha subunits and 1 each beta, gamma, delta subunit. 2 molecules of acetylcholine bind alpha subunit and open the channel for sodium, potassium, and calcium. A deficiency in acetylcholine receptor leads to myasthenia gravis. Ionotropic glutamate receptors bind the neurotransmitter glutamate. They are tetramers. They permit influx of sodium and calcium. They mediate the majority of excitatory synaptic transmission throughout central nervous system. ATP-gated channels open upon binding of ATP. They form trimers. Both the C and N termini are on the intracellular side. A well-known example is cystic fibrosis transmembrane conductance regulator, CFTR. CFTR is a cyclic GMP activated ATP-gated chloride channel. A deficiency in CFTR leads to cystic fibrosis. A lipid-soluble hormone, such as a steroid hormone, penetrates the cell membrane. It binds a nuclear receptor in the cytoplasm. In this case, it is steroid hormone receptor, and displays the heat shock protein. The receptor enters the nucleus and binds the DNA. It acts as a transcription factor and activates transcription of target genes. Leading to increased synthesis of protein. Due to transcription and translation, there's a lag period of 30 minutes to several hours. This slide shows PPAR alpha and PPAR gamma. They are nuclear receptors that are already in the nucleus. After a ligand binds the receptor dimerizes with retinoic X receptor, RXR. PPAR-RXR dimer acts as a transcription factor and controls transcription of target genes. Effects of nuclear receptor agonists can persist for hours or days after plasma concentration is zero. Here is integration with vitamins. RXR can also dimerize with retinoic acid receptor, RAR. Nuclear receptors are classified into several types. Here we will focus on two types, type 1 and type 2. Type 1 nuclear receptors are found in the cytoplasm associated with heat shock proteins. Ligand binding triggers hormone dimerization of the receptor, translocation from the cytoplasm into the nucleus. The receptor binds a specific sequence of DNA known as a hormone response element. An excellent example is steroid hormone receptor that I have shown you earlier. Type 2 nuclear receptors are retained in the nucleus regardless of the ligand binding status. The receptor forms a heterodimer with another protein, and the heterodimer then binds the DNA. A well-known example is PPAR that you have seen earlier as well. Here are some important nuclear receptors. Glucocorticoid receptors are type 1 nuclear receptors. They induce transcription of lipocortin gene, whereby they inhibit phospholipase A2 activity. They also inhibit transcription of the COX-2 gene. Mineralocorticoid receptors regulate expression of proteins involved in reabsorption of ions, especially sodium, in the renal tubules. Retinoid receptors control embryonic development and cell differentiation on the skin. PPAR alpha is a target of fibrates. Fibrates are triacylglycerol lowering drugs. They stimulate beta oxidation of fatty acids. PPAR gamma is a target of glitazones. Glitazones are anti-diabetic drugs. They increase storage of fatty acids in adipocytes, thereby decrease the amount of fatty acids present in circulation. Cells, therefore, depend more on glucose for energy. To make it easier to distinguish these two receptors, alpha is related to TAG. Gamma is related to glucose. Here is integration with regulation of gene expression. PPAR alpha and PPAR gamma are transcription factors that control transcription of genes involved in fatty acid metabolism. Now let's move to part 3. Enzyme ligand receptor is a transmembrane protein that either contains intrinsic enzyme activity on its intracellular domain or associates directly with an intracellular enzyme. Upon ligand binding, a conformational change is transmitted via a transmembrane helix, which activates the enzyme, initiating signaling cascades. Here are some major enzyme ligand receptors. Receptor tyrosine kinase contains intrinsic tyrosine kinase activity. They are the most common enzyme ligand receptors. Examples include insulin receptor, EGF receptor, and FGF receptor. Receptors containing intrinsic serine-threonine kinase activity, such as a BMP receptor. Receptors containing intrinsic granulate cyclase activity, such as atrial natriuretic peptide receptor. Cytokine receptors are closely resembling receptor tyrosine kinase, except the protein tyrosine kinase activity is not intrinsic to the receptor. They are separate enzymes. Well-known examples include growth hormone receptor, erythropoietin receptor, and interferon receptors. Today, we will focus on receptor tyrosine kinase and cytokine receptors. Here is EGF receptor. Binding of the ligand EGF leads to dimerization of the receptor and activation of the intracellular kinase, receptor tyrosine kinase, or RTK. The GRB2 protein binds the RTK via its SH2 domain. This protein also binds SOS via its two SH3 domains. SOS represents son of 7 leads. It is a guanine nucleotide exchange factor, or GAF. RTK activates SOS, which then promotes removal of GDP from RAS. RAS can now bind GTP and become activated. Several SH2-containing proteins have intrinsic enzyme activity that can be recruited and activated by receptor tyrosine kinase. Notable examples include phospholipase C-gamma, RAS-associated GDPase activating protein, or RAS-GAP, phosphatidylinositol 3 kinase, or PI3K. Activated RAS activates the RAF kinase. RAF phosphorylates and activates MEK kinase. MEK then phosphorylates and activates MAP kinase, or MAPK, or ERK. MAP kinase finally phosphorylates and activates transcription factors, including c-MYC and c-FOS. They control cell cycle. To inactivate the RAS signaling, GTPase activating protein GAP binds the RAS and hydrolyzes the GTP. The GDP-bound RAS is inactive. In many cancers, such as melanoma, overactivation of the RAS-MAP kinase pathway is responsible for uncontrolled growth. Therefore, Many compounds that can inhibit steps in the MAP kinase pathway have been used as drugs for chemotherapy. Cytokine receptors use a mechanism very similar to that of receptor tyrosine kinases, except in this case, the protein tyrosine kinase activity is not intrinsic to the receptor molecule. Instead, a separate protein tyrosine kinase from the genus kinase family, or JAK, binds non-covalently to the receptor. Here is a cytokine receptor. After a cytokine binds a cytokine receptor, it dimerizes. JAK, a separate protein tyrosine kinase, binds and phosphorylates the receptor. The receptor then recruits STAT, a transcription factor. JAK then phosphorylates STAT, which dimerizes and moves to the nucleus. Now, let's move to part 4. G-protein coupled receptor is a transmembrane protein that crosses the lipid bilayer 7 times. When a ligand binds to a receptor, it causes a conformational change in the G-protein coupled receptor, which allowed it to act as a guanine nucleotide exchange factor, or GAF. The receptor then activates an associated G protein. Here is the receptor, and here is trimeric G protein comprised of alpha, beta, and gamma subunits. When a ligand binds the receptor is activated. It acts as a guanine nucleotide exchange factor, or GAF. The receptor activates G-alpha by exchanging its bound GDP for GTP. When bound by GTP, G-alpha can then dissociate from beta and gamma subunits to control target protein activity. To terminate the signal, GTP is hydrolyzed to GDP. The alpha subunit with bound GDP forms a trimeric complex with beta and gamma subunits, which is then again associated with the G-protein coupled receptor. G proteins are GTP binding proteins. They participate in reversible GTP-mediated interactions. There are two groups of G proteins. First group is represented by small GTP binding proteins. They act downstream of receptor. Examples include RAS, RAC. They typically mediate receptor tyrosine kinase signaling. Second group is represented by heterotrimeric G proteins. They directly couple to receptor and enzymes. They consist of alpha, beta, and gamma subunits. They are coupled to a G-protein coupled receptor, such as a glucagon receptor and beta-adrenergic receptor. G proteins share some common features. They bind GDP and GTP with high affinity, but adopt different structures depending on the bound nucleotide. GDP bound complex has high affinity for other proteins, affecting their enzyme activity. G proteins possess intrinsic GTPase activity that is usually activated by interaction with a regulatory protein such as GAF. G proteins are typically attached to cell membrane through covalent attachment of various lipids. There are two major targets of heterotrimeric G proteins. One is adenylate cyclase, or AC. Second one is phospholipase C-beta. As shown here, adenylate cyclase generate cyclic AMP. Cyclic AMP then activate protein kinase A. Activated protein kinase A phosphorylates target proteins. Adenylate cyclase is activated by G-alpha S and inhibited by G-alpha I. Phospholipase C-beta cleaves phospholipids at C3 position, giving rise to IP3 and diacylglycerol. Phospholipase C is activated by G-alpha Q. Here is the integration with microbiology. Cholera is an infection of small intestine caused by bacteria Vibrio cholerae. Pertussis is also called the whooping cough. It's a highly contagious bacterial disease caused by Bordetella pertussis. In both cases, G protein-coupled receptor signaling is abnormally enhanced. In these two cases, bacteria produce a specific toxin. Cholera toxin causes ADP ribosylation of G-alpha S. It inhibits the release of GTP from G-alpha S. Therefore, G-alpha S is trapped in active form, which causes a rise in cyclic AMP level. Cyclic AMP regulates secretion of fluid into intestine. A rise in cyclic AMP causes enormous loss of liquid in electrolytes, leading to severe diarrhea. Pertussis toxin causes ADP ribosylation of G-alpha I and inhibits the release of GDP from G-alpha I. Therefore, G-alpha I is trapped in its inactive form and cannot inhibit adenylate cyclase. Now, let's move to part 5. First messengers are extracellular substances that have a receptor on the plasma membrane. Second messengers are intracellular molecules released by the cell in response to exposure to the first messengers. Cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, and calcium are second messengers. The main effector of the cyclic AMP PKA pathway is protein kinase A. It consists of 4 subunits. Here's protein kinase A. 2 regulatory subunits and 2 catalytic subunits. After cyclic AMP binds to the regulatory subunits, the catalytic subunits dissociate and move to the target substrate. One target is CRAB, a transcription factor. PKA phosphorylate CRAB, and phosphorylate CRAB binds the cyclic AMP response element in the enhancer of cyclic AMP responsive genes, including 2 genes in gluconeogenesis, PEPCK and G6PC. PEPCK encodes PEP carboxy kinase. G6PC encodes the catalytic subunit of glucose 6-phosphatase. The cyclic AMP protein kinase A pathway also controls glycogenolysis. Protein kinase A phosphorylates and activates glycogen phosphorylase kinase, and this kinase then phosphorylates and activates glycogen phosphorylase. Protein kinase A also phosphorylates and inactivates glycogen synthase. Many receptors function through protein kinase A, Examples include glucagon receptor, beta-adrenergic receptor, and many more. Second major target of trimeric G protein is phospholipase C-beta. Phospholipase C-beta cleaves PIP2, and here is PIP2. It is associated with cell membrane. Phospholipase C-beta cleave PIP2 and release diacylglycerol and IP3. Diacylglycerol and IP3 are second messengers. Here is integration with complex lipids. PIP2 is a phospholipid, a component of cell membrane. IP3 diffuses through cytoplasm and binds IP3 receptor on ER. And here is ER and this is IP3 receptor. Activation of IP3 receptor releases calcium from the ER. Both diacylglycerol and calcium activate protein kinase C. Receptors that function through phospholipase C calcium protein kinase C pathway include angiotensin II receptor in the vascular smooth muscle, catecholamine alpha receptor, TRH receptor, and gonadotropin-releasing hormone receptor. These receptors can be summarized in one mnemonic: ACTG. A stands for angiotensin II receptor, C stands for catecholamine alpha receptor, T for TRH receptor, and G for gonadotropin-releasing hormone receptor. Second messenger, cyclic GMP, functions through protein kinase G. Cyclic GMP is produced by granulate cyclase. This second messenger is found in only a very few cell types. Nitric oxide activates cytoplasm granulate cyclase, as shown here. Cyclic GMP stimulates cyclic GMP-dependent protein kinase, or protein kinase G. Protein kinase G phosphorylates and activates myosin light chain phosphatase. Dephosphorylation of myosin light chains leads to relaxation of smooth muscle. This action is terminated by a hydrolysis of cyclic GMP and dephosphorylation of myosin light chain phosphatase. Signals mediated by cyclic AMP and cyclic GMP can be attenuated by phosphodiesterases, or PDEs. There are several PDE families. PDE3 degrades cyclic AMP. PDE4 degrades both cyclic AMP and cyclic GMP. PDE5 degrades cyclic GMP. Methyloxanthines such as coffeine and theophylline are PDE3 and 4 inhibitors. In smooth muscle, these inhibitors enhance signals from adrenergic receptors, leading to accumulation of cyclic AMP and vasodilation. Sildenafil is a specific PDE5 inhibitor. It raises cyclic GMP levels in smooth muscle, which cause relaxation of smooth muscle and increase inflow of blood into the spongy tissues of penis, leading to an erection. Now, let's move to part 6. A high level of calcium outside and low level of calcium inside the cell generate a steep calcium gradient. Only slight changes in membrane permeability will result in dramatic changes in calcium concentration in the cytoplasm. A change in calcium concentration alters the activity of calcium-binding proteins and triggers physiological change of the cell. For nerve, cardiac, and smooth muscle cells, calcium can be derived from outside. It may enter the cell through voltage-gated calcium channels, ligand-gated calcium channels, or store-operated channels. Here, I highlight L-type calcium channels. They are long-lasting voltage-gated channels, responsible for normal myocardial and vascular smooth muscle contractility. Intracellular compartment provides calcium for both skeletal and cardiac muscle, although in two different ways. In skeletal muscle, voltage change is sensed by dihydropyridine receptors that are linked to ryanodine receptors in the SR membrane. Ryanodine receptors are calcium channels. Activation of dihydropyridine receptors triggers release of calcium from SR through the ryanodine receptors. The SR provides virtually all the calcium ions for skeletal muscle contraction. In cardiac muscle, calcium entering the cell through the voltage-gated calcium channels activates the ryanodine receptors, triggering the release of calcium from SR. This calcium-induced calcium release is the primary mechanism of activation in cardiac muscle. Intracellular compartment also provide calcium in other non-muscle cells. Calcium release from the ER is regulated by IP3 receptors in the ER membrane. IP3 receptors are typically regulated by G-protein coupled receptors. IP3 is produced through the activity of receptor-activated phospholipase C. IP3 diffuses through the cytoplasm and binds IP3 receptor. IP3 receptors are located in the ER membrane in many tissues. To stop muscle contraction or other calcium-mediated activities, calcium needs to be removed from the cytoplasm. Removal of calcium from the cytoplasm is first achieved by calcium pumps, and then by calcium buffers. They are calcium-binding proteins with a low binding affinity and a high capacity. There are three major pumps, two on the plasma membrane and one on ER or SR membrane. First pump is plasma membrane sodium calcium exchanger, or NCX. It is mainly found in excitable cells such as cardiac and skeletal muscle cells. 3 sodium ions are imported for 1 calcium to be exported. Second pump is plasma membrane calcium ATPase. 2 calcium ions are exported for each ATP molecule hydrolyzed. This pump is regulated by protein kinase A, protein kinase C, and calmodulin. Third pump is SR or ER calcium ATPase. It is the most abundant protein in the SR membrane. Here is integration with pharmacology. digitalis alkaloids, such as digoxin, are used to treat heart failure. They are inhibitors of sodium-potassium ATPase. These drugs make sodium-calcium exchanger less efficient and therefore raise intracellular calcium levels, which causes stronger muscle contraction. Another drug, warfarin, works in a very similar manner. Cefzigagin is a lab reagent that inhibits SRER calcium ATPase, whereby it also raises intracellular calcium concentration. Calcium controls a wide range of cellular processes. Here are some notable examples. Annexin, troponin C, and calmodulin are direct targets of calcium, and they are calcium sensors. Among them, troponin C and calmodulin are EF-HANS proteins. EF-HANS protein is named after the shape created by the E and F alpha helices of the calcium binding domain. Calcium also controls release of neurotransmitters and hormones. Two well-known examples are the release of neurotransmitter into the synaptic cleft and release of insulin from pancreatic beta cells. Today, we will focus on these three proteins. Annexin is also known as lipocortin. Annexins are a family of proteins with a common feature that they interact with membranes in a calcium-dependent manner. They are implicated in regulation of phospholipase A2, cytoskeletal reorganization, and vesicle movement. Annexins have a low affinity for calcium ions, which restrict their action close to the cell membrane. Lipocortins suppress phospholipase A2. Glucocorticoids, such as cortisol, increase synthesis of annexin-1, which inhibits phospholipase A2. This provides a mechanism for how glucocorticoids suppress inflammation. Second protein is troponin. Troponin forms a complex of three regulatory proteins, troponin C, troponin I, and troponin T. And here is a complex made up of troponin C, troponin I, and troponin T. Troponin complex control muscle contraction in skeletal and cardiac muscle, but not smooth muscle. Here is integration with physiology. Troponin I inhibits formation of cross bridge between actin and myosin. Muscle relax. When the calcium level rises in the cytoplasm of muscle cells, troponin C combines with calcium and blocks the action of troponin I. Muscle contracts. The third protein is calmodulin. Calmodulin is ubiquitously expressed. Each calmodulin molecule binds 4 calcium ions. Calmodulin calcium complex is an essential component of many calcium-dependent enzymes. Calmodulin acts through stimulation of either calmodulin-dependent protein kinases or CAM kinases, or protein phosphatases. Calmodulin also activates cyclic GMP phosphodiesterase. Here is a function of calmodulin in muscle cells. Glycogen phosphorylase kinase is a CAM kinase. Calmodulin calcium complex activates glycogen phosphorylase kinase B directly without phosphorylation, which is shown here. This is a function of calmodulin in smooth muscle cells. In smooth muscle cells, when the calcium level is low, caldesmon forms a complex with actin and tropomyosin. Access of myosin to actin is restricted. Muscle relaxes. When the calcium level is high, calmodulin binds caldesmon. Inhibition is lifted. As a result, muscle contracts. Calmodulin also regulates myosin. When the calcium level is low, myosin light chains inhibits actin and myosin interaction. When calcium level is high upon cell signaling, calmodulin activates myosin light chain kinase. This kinase then phosphorylates myosin light chain. Inhibition is lifted. Muscle contracts. Calmodulin also functions in T cells. Here is integration with immunology. When an antigen binds a T cell receptor is activated. Activation of T cell receptor leads to an increase in the calcium level in the cytoplasm of T cell. Calmodulin binds calcium and activates calcineurin. Calcineurin is a protein phosphatase. It removes phosphate group from mFAT-C on serine and threonine. mFAT-C then translocates to nucleus, where it combines with mFAT-N and induces transcription of interleukin-2 gene. Interleukin-2 is a cytokine that promotes division of T cell. Here is integration with pharmacology. Calcineurin is a target of immunosuppressive drugs FK506 and cyclosporin. These drugs form a complex with immunophilins. These proteins compete with calmodulin for binding to calcineurin. As a result, there will be no mFAT-C activation and interleukin-2 induction. This provides a mechanism for immunosuppressive effects of these drugs.